The last lecture covered the non-specific innate host defenses, meaning those defenses that we are born with and that react in exactly the same way each time they see a pathogen. There's no change, there's no increase or decrease in the reaction, and it's not specific. There's no adapted particular receptor to the pathogen. Chapter 18 is dealing with the adaptive specific host defenses. That means these are mechanisms in our bodies that can change in response to being exposed to a pathogen. That there's an initial response and there's a secondary response. And that is how our lifelong immunity to certain pathogens and other antigens works. This slide shows us a polio ward where individuals are in iron lungs to breathe for them because polio causes paralysis. It was an extremely common disease worldwide, including the United States, until a vaccine became available. And this image is from 1953, so not that long ago in the concept of modern medicine. This slide is showing immunological memory. Had to pause so that I could get the screen to actually include the top of the slide. So immunological memory. We have some sort of exposure to a pathogen, whether it's bacteria or a virus or a fungus or a worm or pollen, and that's our initial exposure. It takes some time to build up this immune response that is specific. So our non-specific immune response is involved and will generate things like inflammation. And those cytokines that we saw in the last lecture work not only within the innate immune response, but also trigger responses in the adaptive immune response. And after we recover from whatever pathogen we've been exposed to, then the amount of circulating cytokines in cells decreases. But if we are re-exposed to that same, or even sometimes a very similar pathogen or antigen, our secondary response is much quicker and much stronger. This happens in natural immunity. For many diseases, if you've been exposed, then a second exposure, your adaptive immune response is so strong that you do not come down with any signs and symptoms or the disease. This is also how vaccination works, where you are exposed to an antigen, some small part or an activated portion of that pathogen, and the antigen, without the whole pathogen present, triggers that primary immune response, and that is why after vaccination you might feel lethargic, you might have body aches, you might even have a slight fever, because your immune system is working. Both your innate and adaptive immune systems are trying to function to eliminate that antigen. And then after that response subsides, if you are actually exposed to the illness, your secondary immune response will be rapid and efficient and will eliminate that pathogen without you having to experience the illness. We keep mentioning antigens and antibodies. So antigens, as I mentioned in the previous lecture, is any substance that generates an antibody response. They are typically proteins of some sort. And an entire antigen may have separate components on it called epitopes. And those are the specific places on which an antibody can bind. So an antigen might only have one epitope. It may also have multiple epitopes. And so you might have 3 different antibodies that might all recognize this antigen. antigen. So I mentioned that antigens are usually proteins. They typically have multiple epitopes so that our bodies have more than one way to recognize and eliminate them. And this is showing how we could have 3 different antibodies that would each specifically bind to its epitope and try to trigger those other immune responses. So antibody 1 has a fork shape that recognizes this epitope. Antibody 2 recognizes the conical one, and antibody 3 recognizes a circular one. So antibody 3 cannot recognize the cone or the rectangular epitope. Likewise, the other antibodies, they're each specific to that one epitope. And this binding of antibody to antigen is one way that we activate that non-specific complement pathway. This is the classical activation pathway. This is a cartoon of the generalized structure of an antibody. There are 4 chains, so it is tetrameric. We have 2 long chains that are called the heavy chains, and we have 2 individual light chains that are smaller. The heavy chains are bound covalently to each other with disulfide bonds. So those are covalent between sulfur functional groups. And each light chain is bound on one side of the heavy chain also by disulfide bonds. Because the antibody is protein, proteins are made out of amino acids, and the amino acid cysteine contains sulfur. So there have to be cysteine amino acids in this position where we see the disulfide bonds. 2 heavy, 2 light chains that make this fork-shaped protein. We have the antigen binding site, which is here at the very tips of the fork. And we also have something called the variable region that is just this very tip of both the heavy and light chains. And we have the constant region, meaning does not vary. All of the antibodies are going to have, in that particular category of antibodies, are going to have the exact same amino acids in the same order forming that shape. The only variability is in this antigen binding region. So this variable region is, in addition to the whole light chain, is called the FAB because it's the fragment, that's the F, and the AB is antigen binding. And this portion at the bottom is the FC, the fragment that is always constant. This is an actual molecular model showing the 2 heavy chains and the 2 light chains. It happens to be IgG. This is how we abbreviate the antibody classes. Ig stands for immunoglobulin. So globulin, it's a globy-shaped protein. Immuno, it's acting in our immune systems. And G, there are different types or categories of antibodies. So there's immunoglobulin G, immunoglobulin A, and E, and D, and M. Those are just the categories. They're all immunoglobulins. So here is our table: IgG, M, A, D, and E. 3 of them are monomers, meaning the whole thing is one antibody when we link our chains together. IgM is a pentamer. You need 5 separate little forks to create the entire antibody. And IgA, you need 2. It's a dimer. I will, let's see, I can mark it right now if I use a pen. Here's my pen. Let's see, my pen color. Sure, red is good. What do we need to know? Oh, we need to know if it crosses the placenta. That's an important. If it fixes complement and their functions. You do not need to know the number of antigen binding regions or what percentage they are in the human body. But crossing the placenta is important because that means if a mother has antibodies to a particular pathogen, if they cross the placenta during embryonic and fetal development, then that fetus, when it is born, has temporary protection of those diseases because they have the antibodies from the mother. Antibodies you get from someone else through the placenta or through breast milk or even through injection only circulate for about 6 weeks. So that is why breastfeeding is important, because it continues to supply the antibodies to the infant. None of the other categories of antibodies can cross the placenta. Fixing complement means when you have the antibody bound to antigen, does that trigger that non-specific complement activation to get the C6, 7, 8, and 9 to form the membrane attack complex and destroy the pathogen. And so IgG and IgM are capable of doing that. And then the function, we have several different things down here depending on the antibody, and we'll see some specific examples of those. It's a great one. We have a virus. So how do our antibodies interact with a virus? How do they interact with a toxin? How do they possibly interact with a bacterium? So the process is called neutralization, because if we get the IgG or the IgM antibody bound, then it will neutralize that pathogen because it will trigger other immune responses to eliminate it. Because we only have single little Y-shaped antibodies, in this case, if it's triggering the complement system, these are IgG antibodies. So our antibodies might stick to proteins sticking off of the virus. They would, if they're acting against a toxin, act on the binding portion of it. And in this case, they are binding to the flagella, but there could be proteins on the surface of the bacteria that could also be targeted by antibodies. So we're looking at the Fc. If we think back to our structure of the antibody, the Fc is the constant fragment, the part that is the same in all of the antibodies of a particular class. And macrophages have Fc receptors. That means they will bind to the base of that fork of the antibody so that the antigen binding portions are out and able to stick to antigens. So we see an antibody with its Fc constant region bound to the Fc receptor after the antibody or antigen binding portion of the fork had stuck to some pathogen. So the circulating pathogen is out there, the antibody forks stick to it, and after that has happened, the macrophage can recognize it because the Fc receptor will stick to the antibody's constant region that is bound to the pathogen. The macrophage can engulf it and bring it in. Igm antibodies do a thing called agglutination to cause clumping. Go, get me out of the way. This is showing some species of bacteria. There's various epitopes on its surface, and those pentamer IgMs can bind across more than one bacterium. So this IgM antibody has a specific binding site for the green epitope. This is a different IgM antibody because it is binding to the circular epitope. So an IgM could also, one IgM, because it has 5 forks on it, could have 3 different antibody binding sections. Really no good place to keep me on these slides. So antibodies, we saw, could bind to a pathogen and then the constant region bind to a phagocyte to bring it in. The IgM antigen binding portion could stick to a pathogen and cause it all to clump up, and that's another way it could be eliminated. In this way, we have our good old natural killer cell also has Fc receptors on it, and so that if antibodies have bound to the antigen, the constant region can stick to the Fc receptor, then the natural killer cell will release its perforin and perforate the bacteria and kill it off. So ADCC is antibody dependent cell-mediated cytotoxicity, because in order for this natural killer cell to kill this pathogen, it needed a specific interaction. The antibody bound to the antigens on the bacteria, then the natural killer cell could bind to the antibody. That's what triggered the perforins. We saw the MHC in our innate immunity because all of our nucleated cells have that MHC class 1. That's what is one of the signals that says it is self, so that our natural killer cells can recognize MHC1 and know that is a cell that is belonging. MHC class 2 is only found on macrophages, dendritic cells, and B cells. So we have an alpha 1, 2, and 3 on the MHC class 1 with a single beta 2. The MHC class 2 has different proteins entirely. It still has a place where an antigen can bind, but it's got a beta 1 and 2 and an alpha 1 and 2. So 4 chains for each. The shape of them are different. The proteins are made out of different. The antigens that they can bind are different. So when a phagocyte or a dendritic cell is going to engulf a bacterial cell, we saw that in the previous slide, that it was non-specific, but it can also happen in the context of antigen presentation. That means that the pathogen gets digested in that phagolysosome. So we had that all brought in, and then to trigger a further immune response, the antigens that were left over from the digestion bind to MHC class 2 and get presented, shown, stuck to the outside of that dendritic cell. And then that is a signal to a different type of cell called a T cell that could come and do additional reactions. We still engulfed a bacterial pathogen through phagocytosis. That's in the phagosome. The phagosome fuses with the lysosome. We burst and kill the bacteria. But any of those antigens can bind to MHC class 2 and go on the surface, and then that will show or present that antigen to T cells that will then create memory. There's a T lymphocyte. It's got all kinds of receptors on its surface, and it also increases its surface area to get all sorts of interactions with signals and other cells. We saw in our innate immunity lecture that all of the formed elements, all of the cells in our blood, form in the bone marrow. We're looking at the red bone marrow, forms all of our erythrocytes, which are red blood cells, the leukocytes, which are the white blood cells, and the platelets. And the yellow marrow, the middle, is our adipose tissue, our fat. So the two main categories of cells in the rest of the lecture are T cells and B cells. They originate in our bone marrow, but they mature or finish their differentiation in different places. The T cells mature in the thymus, and the B cells actually finish their maturation in the bone marrow. This is looking at our T cells. It is has two lobes, and it's located just above heart. There's our thymus. And that is where these cells mature, but then they can circulate into our lymphatic system and accumulate in our lymph nodes, which are found throughout our body. Our tonsils are also lymphatic tissue. Our spleen is also part of our immune system. So antibodies are those little forks. They go out and bind to antigens. But T cells will bind also to antigens that are presented when they're stuck to either the major histocompatibility complex 1 or 2. And they're abbreviated TCR for T cell receptor. So they can only bind antigen if it's on a cell with the major histocompatibility complex. So antibodies can bind antigens on cells circulating in the fluid. They can bind antigens anywhere they find them. T cells can only bind antigens that are already stuck to a major histocompatibility complex. And just like our antibodies, they have a variable region, which is where the antigen binds, and they have a constant region that is similar with all T cell receptors. They are connected with a disulfide bond that is covalent, and they are embedded in the plasma membrane of the T cells. The two major classes of T cells are T helper cells, or T sub H, and cytotoxic T cells, T sub C. So unactivated helper T cell, called naive because it has not yet bound to antigen. So a dendritic cell engulfed something, some pathogen, and broke down that pathogen and now has the antigen of that pathogen stuck in, presented on its MHC class 2. Once that happens, a helper T cell receptor will stick to the antigen on the other side of it. And when that happens, it can proliferate, which means divide, and differentiate, meaning change into either a helper type 1 cell, a T helper 2, or a memory cell. This is just making copies to fight against the antigen that's currently present. Some of them are still able to fight against that same antigen, but they will be stored in our lymphatic tissues, and those are the memory ones. They hang out there and don't continue the fight. They're going to be there for the second time that antigen shows up. The cytotoxic T cell, same thing, it's naive because it has not yet bound antigen. There will be some sort of intracellular pathogen that is present. This is not being engulfed necessarily by phagocytosis. And MHC class 1 now has the antigen on its surface. The cytotoxic T cell has a T cell receptor that binds to the other side of that antigen. That activates it to secrete enzymes and perforins to kill that infected cell. So this is not creating memory. This is directly acting when this cytotoxic, which means toxic to cells, when it binds to the antigen, it releases the chemicals that will cause apoptosis, the self-destruction of that cell. So the other molecules that are on those surfaces, T helper cells have CD4, cell determinant 4. The cytotoxic T cells have cell determinant 8, or CD8. This says a T cell because it has CD4. It has to be a T helper cell. So it is recognizing epitopes of antigens on class 2 MHC, and that is going to, in a normal sense, would cause, the proliferation of the T cells and create memory cells. This is saying that's our normal stage. What if we have binding of an antigen by its epitope on a macrophage, but the shape of this is not quite right? It doesn't really fit into the T cell receptor. Even though it has touched it, we're not going to activate this T cell receptor. That's a mismatch. It would have to go off and find the one that does match in order to be activated. Another thing that could happen is something called a super antigen. Doesn't mean it's the better antigen. It means it creates a hyperactive response. And what's missing in this image, we have a T cell, its receptor is bound to the MHC class 2, but there is no antigen in this cleft. No antigen bound into the cleft of MHC2. No antigen is bound in the antigen binding site of the T cell receptor. Instead, a super antigen sort of glues the MHC class 2 and the T cell receptor together. And that causes a non-specific, uncontrolled activation. It releases cytokines that trigger other T cells. It might trigger cytotoxic T cells. It will trigger B cells to divide. And we haven't really had a specific interaction, and we get non-specific inflammation. Here's our antibody again, where we have the variable region and the constant region, the antigen binding sites being at the tip, and the variable regions on a single cell will always bind the same antigen. So one antibody, we're only going to have one antigen that these receptors can bind to. And these are made based on the instructions in our DNA. Right? It is a protein, or it's a series of proteins that connect together into one giant protein. Right? This would be its quaternary structure. So our DNA contains multiple sections for creating antibodies, and depending on which combinations of those genes will determine which combinations of antibodies. And we have enough variations of those genes to create antibodies against pretty much anything that could show up. All right, get me in the middle here. So T, meaning T cell, independent antigens have lots of those repeating epitopes that get the B cell to recognize it and activate without any T cells. Here is our pathogenic bacterium. The B cell has antibodies that are binding to it, and that is triggering cytokines that will activate itself, and it will secrete those Igm antibodies. And then it will differentiate into secreting those Igm antibodies. We could also have T cell dependent activation of B cells. In this case, we have the B cell. It's stuck on some sort of antigen that's on a bacterium, but it doesn't really get activated. The B cell is not activated until we also have the B cell present the antigen with its MHC class 2. That triggers the T cell to send out those cytokines that cause the B cell to proliferate, and we will get the plasma cells, meaning they secrete the antibodies out into the plasma and they can circulate anywhere to fight the pathogen. And we also create the memory B cells that keep the antibodies attached to them so that in the case of a second exposure, they are ready to proliferate and become plasma cells. We saw the primary and secondary responses in one of the first slides. This is showing that the primary and secondary responses can differ depending on the type of antibody made. The IgM primary and secondary responses, the level is about the same even after secondary antibody exposure or antigen exposure. The IgG, however, is higher than IgM in the initial response and dramatically higher in the secondary response. It also lasts longer. IgM comes on moderately and wanes pretty quickly. We have a peak, we have antigen exposure, the peak is around 14 days, and it drops off about 18 days. Takes IgG a little longer to show up, around 20 days, and it's also in a couple of days it wanes. But with the secondary response, here it is, within 15 days, we're at like triple the level of the IgMs, and it circulates much longer in the system. In adaptive immunity, we have to acquire this. We have B cells and T cells, but until they become activated by a specific antigen, then they're not really working. So we have to acquire, be exposed to antigen in order for that to work. So how do we acquire our immunity? We can acquire it naturally. Naturally by a passive immunity, meaning we get the antibodies from someone else. So breast milk or through the placenta. We could also get passive immunity, that's something for passive natural immunity. Active natural immunity means you get sick. You pass through the illness, you manage to recover, and you get immunity to it. And that is not what happens in every case. The reason why we have a different flu vaccine every year is that, you might be immune to that particular H and N combination, but not to the next one. And other diseases just don't create enough of an immune response for you to be immune to it throughout your life. We can artificially acquire immunity, both passively and actively, harvesting antibodies from someone else, meaning inject them. So that is one of the proposed COVID-19 treatments, that people who recover from COVID-19, at least for a while, have circulating antibodies. And so if they donate blood, the plasma can be separated from that, and that plasma contains the antibodies, and those antibodies can be injected into someone suffering from COVID to help them recover. Mixed success with that, and we don't know how long those antibodies last, even in the person who got them through natural active immunity. The first hepatitis A vaccine I got was actually passive artificial immunity. I was going on a trip and I didn't find out enough in advance to get the actual vaccine, and so I was given hepatitis A immunoglobulin, somebody else's purified antibodies. Once I returned, I completed my hepatitis vaccine. Artificially acquired active immunity is through actual vaccination, and vaccines contain just a portion of a pathogen. Either it's some combination of its antigens. In the case of viral vaccines, it can be nucleic acid, or it can be inactivated viruses. And then your body will create that primary immune response, so that if you are exposed to the whole and intact pathogen in the future, your secondary immune response will react quickly and strongly enough to prevent you from succumbing to illness. Variolation was an ancient technique exposing patients to small amounts of a disease in order to create immunity, and that was also done with smallpox with the initial vaccines, exposing individuals to cowpox, which is a related illness, and that created immunity. So there's cowpox. If you get infected with cowpox, the lesions are pretty terrible and painful. They cause inflammation, but it's not deadly. However, the immune response to cowpox, it is a very closely related illness, will make a person immune to smallpox.